looking forward to tell you all about Arcturus and how we combine robotics and machine learning for accelerated drug discovery. So a little bit of background about our purpose here is that the need for new medicines is really endless. As we move from the old era of really horrible toxic chemotherapies into the era of targeted medicines, there's an endless requirement for better and improved drugs to treat all types of disease. And with an aging population, these problems become even more significant. So the fact that 1 in 2 of us will be diagnosed with cancer at one point in our lives is quite terrifying. So we really need to be working continuously to develop drugs and to develop drugs in better ways. The drug discovery process overall is lengthy and costly. It hasn't been improving, as I'll come to in a second. So the origin is often understanding the basic biology, understanding the disease and the etiology of the symptoms and the drivers of the disease. The conclusion of that point is a target hypothesis, the belief that a certain protein or protein dysregulation is driving the disease, or from there you have an angle from which to try and target that protein and address the disease biology to rectify the symptoms. So once we've decided on a target, we need to move into finding novel chemical matter. This is the hit discovery phase. So conventionally, people would have done high throughput screening. Increasingly, this is reduced to in silico approaches, which are much more cost effective than computational chemistry and virtual screening instead. From that point, you have the origins of potential chemical matter, which can be developed finally towards a drug that's suitable for patients. And along that journey, you'll evolve that chemical structure to gain all of the desired properties in terms of efficacy, potency, safety, and so forth. So it's a really long, complex journey, ultimately optimizing many, many parameters of the molecule simultaneously. So it's a complex process. And it's no surprise then perhaps that failure rates are quite high. We believe in a certain drug, but we only find out once it goes into patients that it may be non-effective or suffers from toxicity, and then it's going to drop out of massive costs. So the total price, if you like, to develop and bring a new drug to market is in excess of $2 billion, and it's a process that takes 10 to 14 years. Typically, this is driven by sort of 3 parties, 3 different sectors of the industry. In academia, people will be exploring and understanding new biology, gaining a deeper and better understanding of the disease. This is often where novel target hypotheses originate. These are most commonly picked up by biotech companies, ranging from 5 to 500 people in size, and they'll be driving forward that early understanding, validating the target, gaining confidence in this approach to treating the disease, and subsequently generating and progressing chemical matter. The pharma is kind of the behemoth of the entire ecosystem. These guys will be watching the biotechs and picking up those advanced molecules at a suitable point, in licensing them, acquiring the biotech, or by other means gaining access to progressing those molecules. And it's the pharma companies that have the sheer scale and access to capital to be able to launch clinical trials and also to be able to survive a program that fails, which would typically be more than a biotech could bear. So here are the kind of traditional organizations laid out. So with the exception of high throughput screening, there's a relatively low degree of automation. And as we're about to come onto, traditionally, this is fairly scarce in terms of computational approaches. It's more of a recent evolution, particularly the evolution towards machine learning, deep learning, and so forth. I mentioned that pharma productivity is in decline, and this is a rather tongue-in-cheek trend known as ERIMs Law. It's the inverse of Moore's Law, which we find in the computing chip industry, whereby chips become more and more powerful. Conversely, in pharma, productivity is actually still very reliably in decline, despite the advent of big data, genomics. None of these have really bucked the trend, and so this is a real problem. It's getting more and more expensive to generate new drugs, which is the opposite of what we need right now. As we move towards more targeted therapies for specific cancer types and so forth, we actually desperately need that drugs can be quicker, cheaper, and delivered more successfully. So these 95% failure rates, the escalating costs, are something that's unbearable for our healthcare systems of tomorrow. Why do we think this is happening? Well, if we look at the data that drug discovery is often founded upon, it's quite poor. So a landmark study took a number of academic publications considered very prominent in the space of oncology, and scientists independently would aim to reproduce and find, to see if they could get the same findings by following the same protocols. And in almost 90% of cases, they found this was not the case. So these academic publications, where we have the origin of the target hypothesis, actually are too often flawed, unreliable, and this results in an estimated $28 billion in wasted R&D funding per year. So it's a really drastic problem. Other scientists cannot find the same results in their own hands when repeating others' results. We've also seen that the labs themselves have not considerably changed in decades. There are certainly better microscopes available, but the lab operation, as many of you will know, still involves a lot of human input and manual labor, doing processes which we would consider robotic, transferring small levels of liquid from A to B and back again. Extremely laborious. It's not a great use of human intellectual power, and it's a process which is very error prone and inaccurate. At the same time, the way that we generate data and capture data is pretty archaic, so it won't be a surprise to anyone. They'll be familiar with data sets being generated, captured on a USB stick, moved to a spreadsheet, all of these data in silos, totally different data structures, lacking data annotation, lacking labeling, lacking the context of the experiment from which the data came. And as a result, we're not able to leverage the full value of the data because it's flawed, it's fragmented, and it's generated in a way that is inconsistent. And finally, as I just mentioned as well, the scientists, what should we really be doing with our brains and with our time? We don't believe that the best use of a scientist is to sit behind the bench transferring liquids for 8 hours a day, whether that be cell culture, synthetic chemistry, or in vitro biology. It's just not the way to spend your time. Every day there must be 100 publications coming out in your field, where ideally you could spend that time reading, hypothesizing, analyzing your data, and truly driving forward the scientific process. So we said lab research has not kept pace with innovation. This is slightly provocative. If we look at the labs of the 1960s, they're fundamentally the same as the labs of today. Better instrumentation, good experimental processes, the operations, the data capture protocols, still involving paperwork and extremely outdated processes. And this is in stark contrast to other industries which have taken huge leaps forward thanks to the advent of the Internet of Things, digital analytics, robotics, cloud data computing. So yeah, we see that there's a huge room for change and improvement available to the lab environment. So what have we seen over the past 5 years? There's been an explosion and a shift towards application of advanced data science techniques, including machine learning, applied to drug discovery. And there are very prominent companies now becoming kind of household names, such as in silico, Atomwise, Benevolent. Everyone must have heard of Exscientia in the past year, who have gone on to do great things, raise a lot of money, and even IPO in the recent months. So the ecosystem is evolving, and computational approaches are starting not only to have an impact on drug discovery, but also to be commonplace, recognized, integral to the entire process. The explosion that we've seen, this is data that I pulled from Crunchbase. I'm not quite sure why there's a gap around 2005. I think that's just a glitch in the recording, but is a clear trend for the increase in machine learning and AI as applied to drug discovery. And we think the time is coming now where this increased effort and focus and emphasis on machine learning approaches is really starting to settle in and starting to take a real impact. So my basic sort of simplification of this is that where there is data, there will be an opportunity for machine learning approaches. And therefore, it's no surprise that machine learning and AI is being applied at every single step along the drug discovery pipeline, from the very earliest stages of identifying new targets, such as benevolent AI, through to designing and developing molecules, for example, in silico medicine, all through the late stages of clinical trial data and monitoring and beyond. So it's really an interesting time. If you're involved in data science, you can certainly find an interesting position in the life sciences too. Where we at Arcturus are most interested is in the process of identifying and developing chemical matter that's suitable for drugs. So from the process of hit generation, hit to lead, and lead optimization, our platform generates data and can make use of machine learning approaches to really improve these processes. Perhaps the sort of landmark beginning of this entire area, this field, was a very provocative publication by In Silico Medicine, led here by Alex Javronkov. And they reported that deep learning approaches had enabled a massively accelerated process of identifying novel kinase inhibitors of the DDR1 kinase. And whilst this really split opinion in the industry, it certainly catalyzed a lot of interest and got people to focus on this area, to discuss it and to move it forward. To kind of summarize their approach, they were using a lot of publicly available data sets to make the first sort of models. Their approach is very heavy on generative adversarial networks. And you can see here how they generated a whole array of novel inhibitors, which exhibited very favorable properties. So you can't bypass the real world chemical synthesis and the real world lab testing, but their computational approaches to the design and development of the molecules supposedly shrunk down a process which would have taken up to 2 years to just over 2 months. And that is a drastic shift. It really caught people's attention. There was some critique and some pushback on the validity of this because of the similarity of the molecules to other reported molecules, but nonetheless, they were technically novel. So whichever way you view this publication, I see it as a landmark in the beginning of machine learning applied to drug design. And subsequently, as I said, I think this industry is really settling in. The big names in machine learning drug discovery are closing deals with the big conventional names in pharma and biotech. These are serious deals and serious amount of money being thrown around, which I think recognizes the appreciation of machine learning and its potential impact for drug discovery. So to complete the evolution, in parallel to the explosion of machine learning approaches, there's also been a trend towards increasing automation, not only in drug discovery, but also synthetic biology, and the origin of a new concept called the cloud laboratory. This includes companies like Emerald Cloud Laboratories, Transcriptic, now called Stratios. And I think what we're seeing is that there's a now convergence and a recognition that combining the enhanced approaches towards data science, including machine learning, with very structured data generation that stems from robotic experimentation, creates a new opportunity. And this is where Arcturus and other companies like Recursion and Incitro sit, where we have this synergistic coupling of robotic data generation capabilities with machine learning technologies in order to leverage enhanced data quality, enhanced analytics to make better decisions throughout the drug discovery process. So what do we see? The labs of old that have not changed in hundreds of years. Sorry, hundreds is a bit of an exaggeration there. But now we have the Unisys Platform, which is developed by Arcturus. And this is an end-to-end automation platform. What we mean by that is we automate the experimental process and we automate the data analysis. So all of the resulting primary data, metadata is lifted from the platform into the cloud, pushed through automated analytical pipelines, and the resulting data and analyses are ingested by machine learning models, which allow us to accelerate the drug discovery process. So this is a leap change step forward compared with the processes of old, which would be highly non-digitized, highly fragmented, and extremely slow and unreliable, as shown in the beginning. So to kind of exemplify this, not only does robotic data generation result in consistency and reproducibility, but we can actually deal with, for example, volumes of liquid which are 1 million times smaller than a human can physically deal with, which allows us to design completely different types of experiments and unlock different depth of data. So we view this as the picture shows. Conventional drug discovery is dealing with low resolution data, but thanks to the Unisys automation platform, we can really bring a high definition in data, which allows you to confidently discriminate between molecules, which would otherwise be more or less bucketed into the same group. So with this sophistication and greater depth and precision of data generation, we're able to give a competitive enhancement to any machine learning approach. Because of course, in machine learning, garbage in equals garbage out. So it's really important to us that we're able to feed our models with the highest quality of data in terms of precision, depth, consistency, structure, and annotation. So we generate over 100 more data points per assay, and that gives us a real edge that allows our models to be more predictive in their capabilities. So what's important? To cut the costs of drug discovery is important. To cut the timelines of drug discovery is important. But in my opinion, there's nothing as important as increasing our success rates. Currently, they've kind of stalled at around 90%, 95% failure for the last few years. And we think that's something that really needs to change. So our goal is not only to kind of accelerate and reduce the cost, be more cost effective, but ultimately to generate molecules which have a better chance of success when entering the clinic. Some people kind of overlook the fact that once you get to a clinical trial, your fate is already sealed. Your fate is tied to that molecule and it doesn't change in the clinical trial. So the crucial thing to get right is to make sure that you're able to design the very best molecule to go into patients to reduce the chances of failure due to lack of efficacy or toxicity and so forth. So this is really where we're focused. Here's a little highlight exemplifying some of the difference in data. On the left-hand side, you have what we'd call a traditional serial dilution approach. You take a high concentration of a drug and you dilute it over a series of dilutions to create a dose range. And then that is tested in your assay of choice, be it biochemical or cell-based, to understand the relative potency of the molecule. It's a very crude but effective experiment. And this is used routinely in companies large and small and in pharma. But in our experience, it's very prone to false negatives. It can overlook the temporal aspect, and the lack of resolution provides a level of data which is just insufficient for high quality machine learning approaches. So our version of this is to take much, much greater data density, to take the full temporal and kinetic aspects, the selectivity aspects, the mechanistic aspects, and all of the associated metadata. And the resulting data package is much richer, much more informative and contextualized, and allows us to do much more sophisticated machine learning approaches with it. And this similar philosophy is applied for us across cell biology, biochemistry, biophysics, and beyond, allowing us to apply smarter data science approaches from hit ID all the way through to late candidate selection. So we really believe that robotics and machine learning complement one another. The robotics provide the consistency, the precision, and the reliability in the data generation. And the quality of data is absolutely crucial for machine learning. So if you want to have high quality machine learning approaches, we believe that robotic data generation is the way to go. Our goal for the future really is to move towards closed loop discovery. This is something that's, for us, still in the research domain. We recently published a collaboration with IBM. We're developing automated chemical synthesis approaches. But without that, the chemistry aspect in terms of synthesis is still very much the domain of manual laboratories, the human workforce. The concept of the closed loop laboratory is to connect the entire design, make, test, analyze process, which is the iterative cycle at the heart of drug discovery. So in terms of design, we'd be referring to generative machine learning models. The make would be substituted for robotic chemical synthesis. The test and the analyze is already the domain of Arcturus's automated platform Unisys. And coupling these together gives you this closed loop concept whereby the data output of each step is fed directly and seamlessly into the next step, allowing you to leap forward in chemical space and to explore chemical space far more effectively and far more creatively than when constrained by human chemical synthesis and human medicinal chemistry for the design component. So feeding all of that data that's generated by the Arcturus platform into machine learning models allows you to make sense of not just potency, but simultaneously the toxicity, the solubility, and all other parameters at the same time. So we don't spend 30 cycles developing a potent molecule only to find that it's toxic. Instead, we're able to develop that multidimensional optimization simultaneously from the very beginning. So that's our vision for the future. And we know that there are many emerging technologies that can plug into that. So we're very interested to speak with people with machine learning capabilities, anyone working in automated chemical synthesis, both ligand and structure-based data science approaches, as well as conventional quantitative structure activity relationships, and of course, bio and cheminformatics. So we really see this as a shift for drug discovery. In the same way that once upon a time, to build a tech company, you'd have to invest a huge amount of capital into on-premises computing servers. Now that's been transformed thanks to AWS and other cloud computing services. So to start a tech company, you can now spin up top-end computing power just from your laptop and an internet connection. Well, we would like to see the same transformation for biotech, where accessing Arcturus gives you the full laboratory, the full data science, the full machine learning suite of technologies required to identify and drive forward drug discovery programs. So on that note, I'd like to thank you all very much for your attention and to highlight that we are hiring at the moment. We're always happy to get to know interested people, and I look forward to hearing any questions. Thank you.